first of all, what is molecular docking? Basically, it is a tool which is widely used in computer-assisted drug design, and its aim is to predict the predominant binding modes of a ligand with the protein of the drug target, that is the protein, right? So it can be used to perform virtual screening of large libraries, compounds, and rank the results and propose the hypothesis of how ligands inhibit the target. So there are terminologies which are associated, such as receptor, which corresponds to the protein or target, and the ligand, which is a small molecule or the drug which you are looking for the binding to the drug target. So application of molecular docking, they include hit identifications. And what is hit identification, or what is a hit molecule? That is a molecule which you obtain after a screening process, or a potential drug molecule which you obtain rationally from the wet lab experiments. Then is the lead optimization. Once you have a hit or some compound which is known to inhibit a particular a drug target or a protein, so you can optimize the lead by using computational techniques such as molecular docking to further see its binding properties with the protein at the atomistic level. Docking can be used to study drug DNA interaction, protein-protein interactions, or even protein-DNA interactions. So I hope everybody's familiar with the lock and key hypothesis for the enzyme and substrate binding. So molecular docking works in the similar fashion. So you have a drug target, right? That is the protein, and a ligand, or maybe a library of ligands. So what we are going to do is check the binding of this ligand with the drug target by using computational molecular docking approach, and find out the stability of this complex, basically in terms of binding energy calculations. And there are so many softwares which can do molecular docking studies, and most commonly used are the ones which are listed here, and the freely available one is the Autodoc, which we are going to use in this particular exercise. So what inspires the molecular docking process, right? So this is just a computationally, we are trying to mimic the binding of the ligands to its protein, right? So as you know that a enzyme binding to the protein, it has an equation which is at equilibrium. That is, enzyme once bind to its substrate or inhibitor, it forms a enzyme inhibitor complex, right? A enzyme in aqueous solution and an inhibitor or substrate in aqueous solution, and it is the complex which it is forming, right? So every ligand which is binding with the drug target, it has some binding energy, which is often defined as the amount of energy needed to remove the particular ligand from the active site. So this is the inspiration behind the molecular docking process. So how it is used in so-called structure-based drug design is that you have to have the protein structure, right? Often proteins are the drug targets. That means in particular disease, it is the protein which is either overexpressed or has been identified as potential drug targets. So we need to know the protein structure for conducting molecular docking. So this can come from structural biological experiments such as NMR, X-ray crystallography, electron microscopy, or if you don't have the experimental structure, so we can conduct some computational methods to predict the 3D structures such as homology modeling. So once you have protein structure, so other thing which is required is the ligand. So you can download the ligand structure and computationally see the docking of both the molecules, that is ligand and the protein, and further send it for the evaluations in the laboratory scale and find out the potential lead compounds from the in silico as well as followed by the in vitro experimentations. So another term which is popular is rigid docking and flexible docking. So what is rigid docking? Basically, it doesn't allow any movement of the either protein or ligand. That means both the things are fixed, right? So you have a single structure for both the protein and the ligand. And in flexible docking, you may induce some sort of flexibility either to the ligand or the protein or both. So what we will be doing is some kind of semi-flexible docking in which often our protein is a rigid structure, but the molecule is moving, right? So basically for a particular ligand molecule which we are docking, we generate a multiple conformations. That is number of structures for the same molecule we will be generating and seeing how well it is binding to the target protein, right? So for achieving this, we have something called as search algorithms. Another term which is very famous is blind docking. So molecular docking is a rational approach where you have to define the binding site on the protein, right? Or uh, for example, in this particular case, this is your target protein, which has several binding sites. So which of these binding site is important? That is the work of a computational scientist. First, identify the binding site. For example, if you have a enzyme whose substrate binding properties or amino acids are known, we can go and explore that binding site and we can specify that binding site as on the right hand side. So it has one spot where the ligand will bind. So we can define our grid here. That means the docking calculations will take place only in this grid. So other molecules, other atoms in the protein will be ignored and the docking process will be very specific on a particular site and will involve ligand or protein atoms which are present in this particular grid. In blind docking, if we don't know which is our binding site, but molecule may bind to that protein, so we can enlarge the size of the grid so that it includes the whole protein. It will take some time. It will be exhaustive process because so many number of atoms will be involved. So and can be non-specific binding. So you have to rationally find out which binding site will be preferred in the molecular docking, right? So often we are in this exercise we'll be proceeding with a specific binding site exercise. So what are the components of molecular docking? There are two important components. One is search algorithm, which identifies the possible conformation or the poses. Or you can say if you have single ligand and you can generate multiple poses for a particular ligand. So how that ligand will be modified to generate new structures, that depends on the search algorithm. And second one is so this search algorithm will generate so-called as conformers. So every structure which is generated by the search algorithm is called as a conformer. And these conformers should be further bound to the target protein structure. And they must be scored in order to get some binding conformation, right? So this scoring will be in terms of binding energy score, which is often referred to as binding affinity. So the first thing is search algorithm, which generates multiple ligand structures. And second is scoring function, which scores those binding conformation at the protein binding site. right? So these are two important uh, 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 components of the molecular docking. So there are several type of search functions and scoring functions. Basically, you have shape matching where uh, the ligand is varied according to the shape of the protein, right? There is systematic search. That means a ligand is varied, or the structure of the ligand is rotated at every degree of freedom. That is, the number of bonds which are present in the ligand will be rotated one by one systematically to obtain multiple conformation. As you can see one on the right hand side, that is clustered ligand. So basically this is the same ligand which is having multiple structures. So this is the work of a systemic uh, search, which basically performs rotations around every bond here. So more popular is the stochastic algorithm, that is generating multiple conformations by random seeding. That means it doesn't do the systematic search, because systematic search, if the molecule is large, so there are more number of degree of freedom. So the search, it becomes very exhaustive. So stochastic algorithms, they take lesser time to optimize the ligand and performs the search for the ligand conformations at a much more faster rate. So the program which we are going to use, Autodoc, it also uses a stochastic genetic algorithm. On the other hand, scoring function, which will give you the binding energy score, they can be different types, such as force field based. So we'll be studying force field in the coming lecture, that is tomorrow, um, and which basically calculates the energy of the system based on bonded and non-bonded terms. And then you have empirical scoring function. Basically here we have some uh, experimental binding energies which are calculated, and the unknown binding energies will be calculated based on those empirical values. So knowledge based is where we have the known binding energies. So basically we try to fit that model onto those knowledge based binding energies. And consensus means utilization of all the possible scoring functions to generate consensus scores. So here you can see there are different docking algorithms which use different type of scoring functions. So Autodoc, it basically uses the empirical scoring function. So what is the genetic algorithm which performs the search for the conformations of the ligand? So basically it is an algorithm which is based on the Darwin's theory, or you can say inspired from Darwin's theory of evolution. So we know that Darwin's theory, that uh, it suggests the survival of the fittest. So we'll see how it can be implemented here. So basically degrees of freedom of the ligand are encoded as binary strings, which are genes, right? So what are genes? Genes are degrees of freedom. For example, in this example molecule, you can see some arrows which corresponds to the bonds which can rotate, right? So after rotating each bond, there will be some changes in the structure, and that will be a completely new structure. So as many as bonds are there, so as many as genes are present in the system, right? Secondly, these genes make up the chromosome which actually represent the pose of the ligand. So altogether, these genes will correspond to the single molecule which we are checking here. And then we have mutation and crossover, which are two kind of genetic operators in genetic algorithm. So basically mutation makes a random change to the gene. That is, mutation is nothing but a rotation of a bond at a particular time. And then crossover is the exchange of genes between two chromosomes. That means a particular type of conformation of a one bond will be transferred to the other ligand molecule. So there are these mutation and crossover exchange events which take place in the ligand, and every time there is a change, there will be a new ligand structure which will be generated, right? So now this is the new generation. That means this must be tested for the hypothesis, that is survival of the fittest. So every single change will be scored by a scoring function, right? So this scoring function um, will score the particular pose, and it will decide that whether it is exceeding a threshold, usually in terms of the potential energy. That means if you are starting with a particular conformation of the ligand and you are making changes into this ligand, so whether the new ligand has lower energy or higher energy than that particular molecule, it will depend, right? So if the molecule has lower energy, then that particular ligand will be retained, otherwise it will be rejected. That means only the fittest will be surviving, right? And this will define the next generation, and this can be done to the next generation also. So there can be multiple ligand conformations in this way, and only the ligands which are surviving, they will be kept. So in this way, there will be multiple conformations which can be further tested for the binding energy calculations. So how the scoring is done? This is important that you might have heard that molecular docking, it results in a binding pose and it will generate some binding energy score. So how those binding energy scores are obtained, right? So especially in autodock, they use the semi-empirical free energy force field to evaluate the conformations during docking simulations. And these parameters, they they are uh, they are basically trained on known protein inhibitor complexes for which inhibition constants are known. So basically, there are known binding affinity values uh, which are there, and based on those binding energy values, or in this case, Ki represents the concentration of the drug, which is required to occupy 50% of those receptors. So basically, we have some known binding affinities based on which they model uh, this force field. So this is called as the empirical. You have experimental values, and whenever a new conformation appears, it will, based on those empirical values, it will try to model the binding energy score for the new conformation, right? So let's see how this is calculated mathematically. So to calculate binding energy, there are 6 different interactions which are measured for a molecule, for a small molecule and the protein molecule, right? So basically, there will be an unbound state and a bound state. In unbound state, this is the ligand which is yet not bound to the binding site, and this is the protein which is present before docking, right? And these are two amino acids, representative amino acid in this particular picture. So in bound state, here you can see the ligand is bound into the active site, and there might be slight change in the conformation of the protein residues, right? So this is the bound state. So these are two states, and their values will be used for scoring the binding of this particular ligand. So in unbound state, there are different three different terms here, 1, 2, 3, which are shown in gray lines. So basically ligand unbound intermolecular, that is within the ligand there are two atoms, or this is true for every particular atom. So within two atoms, what are the interactions, right? That is, what is the potential energy between two atoms? Second one, within the unbound between the atoms of the proteins, what are the interaction energy? And then in the unbound, what is the interaction energy between the ligand and the protein? These are three terms. And at the bound state, between the ligand and the protein, and between the ligand and between the protein. These are six terms which are used. So they are put into this beautiful equation here, that is delta G, that is the binding energy of this ligand binding here. So this is the equation where that is the potential energy of the bound term ligand to ligand, that is this one here, then unbound of ligand to ligand, right? So, and so on, right? So basically these six terms are put into this particular equation. So let's see further how this V or potential energies are measured. We know that this is a non-bond interaction. There is no covalent interaction in this case. So these will be non-bond interactions. So there are two ways that these interaction can be calculated. One is van der Waals and another one is electrostatic term. So van der Waals is represented by 12-6 potential, that is Lennard Jones potential. So this is the equation to measure that. It basically contains one repulsive term and one attractive term, right? That means when two atoms, whether they are close enough to, and what is the optimal distance at which they would like to stay. So basically this equation represents that form. And then is the electrostatic term. That is, what are the charged properties of the atoms. So this is basically a representation of Coulomb's electrostatic law, right? So where you have a charge of I and charge of J. Basically I and J are two atoms in this particular case. And these terms will be calculated for all the atoms which are present in the system, right? Ligand and protein atoms. Apart from that, there can be different scoring functions which may have different terms. So these two are essential terms, and these can be there and cannot be there. One is hydrogen bond term, which is very similar to the van der Waals Lennard Jones potential, but here we have values of 12 and 10. And this dissolution terms, which basically corresponds to the volume of the atoms in a particular system. So once you use these equations and put into the this particular equation, so you'll get the binding energy, that is delta G. So let's see what are the practical steps which are involved in molecular docking. So very first one is preparation of molecules, right? So preparation of receptor protein molecule and the ligand molecule. Second one is specify the active site. So once you have prepared the molecules, you should know on the protein where we want to bind the ligand. So this active site, you can obtain the information either from the literature, go back to the literature, read about your protein and find out which are the important amino acid, mark them and note down and see in the visualizer where they are located on the protein structure. Second one is, if your protein structure is having a co-crystal, that is already a protein inhibitor complex is available in PDB database. So you can use that information where that particular molecule is bound, and you can use that information as a binding site information. Third one is, if you have protein structure but you don't have enough literature or inhibitor bound conformation of the protein. So you need to predict where the binding site is. So there are various prediction um, programs such as CAST-P and others which are available, which can predict the binding site of your protein. So once you know the binding site of your protein, you can proceed with building or the ligand molecules. So you need to download the chemicals or small molecules from chemical databases, right? And then assess the docking parameters. So in this case, you'll be learning about how to set the grid, and second one is how to set the run parameters. And finally, run the molecular docking experiment using these parameters. And then finally, analyze the results using DLG file, Autodoc tools, and Discovery Studio visualizers. Three type of methods we will study in this case. So there'll be different type of files which you need to understand what are the file types. So there is PDB file which refers to protein data bank, PDBQT, protein data bank, partial charge and atom type format. This is very specific to the Autodoc program. And GPF, that is grid parameter file, LG grid log file, DPF docking parameter file, and DLG, that is docking log file. So these are some of the file formats which you need to understand. So what are the softwares which are used or required for this exercise? You'll need MGL tools basically for preparation of the molecules, Autodoc for the docking exercise, UCSF camera, and Discovery Studio. They can be used as visualizers as well as preparation of molecules. And then the actual protein file, which you can download from RCSB website, that is PDB Protein Data Bank website. So we'll be using the DPP for dipeptidyl peptidase 4 enzyme, which is implicated in type 2 diabetes. And the PDB ID is 2RGU. So we'll use this PDB ID to for molecular docking. And we'll be testing one established drug, that is linagliptin, which is a drug which is very specifically binding to this DPP4, and it is a known drug of this particular enzyme. So we'll study the interaction of linagliptin with this particular protein. So finally, in this after this exercise, you should be able to generate these type of images. So first one is 3D pose, that is how this pink colored ligand is binding at the protein binding site, that is the here. And secondly, the same ligand, how many or what type of interactions this ligand is forming with the protein in terms of a 2D plot, 2D interactions plot. So here is the same ligand you can see in pink color, which is bound in 3D conformation here. And then these are the residues which are interacting with this particular ligand. So we'll study and we'll try to generate these images at the end. So it's time for a practical demonstration. So till now, if you have any questions, you can ask, or do we have a separate question answer session? Can you confirm?
Good evening, sir.
Yes, good evening.
Sir, I have a question related to the ligand molecule. Actually, I'm having a ligand. I just want to do the docking, but
Right.
only 2D model of that ligand molecule is present in the PubChem.
Yes.
So for docking, I need a PDB file for that, but I'm just getting SDF file for that. Can
All
you
right.
please suggest further what I should proceed with?
Yeah, sure. So I'll be showing you step by process. So basically, if you're, I mean, how you are preparing the ligand, if you can just tell me. You are not unable to open the ligand only, right?
Yes, sir. I'm just getting the SDF file from PubChem, and from other side, I'm not getting basically that molecule, like any information related to that molecule. But my
Ah,
enzyme is particularly binding to that single molecule only.
okay, that's great. So you can open that file in Discovery Studio Visualizer, or basically if some software, preparation software. So I think UCSF Chimera, which we are going to use for preparation, should recognize your molecule, right? SDF file format. So let's see how does it work. If you still have question, then you can let me know. Okay, we'll go to demonstration.
Sir, actually, I have tried OpenPabble for converting it to the PDB section, like PDB file, but it's showing some stereochemistry error, which I'm unable to rectify.
Okay, so I hope we need to generate a proper 3D conformer for that using UCSF Chimera. So let's see it practically. Okay.
Okay, sir.
Yeah, so let me check the chat again. Okay, so in this particular exercise, we'll be doing single protein ligand docking. So it is possible that you can check multiple ligands. And usually, if you are using Autodoc program, so there will be a single molecule and protein binding only. But if you use molecule Autodoc vina, it can contain. You have to basically uh, generate a script file where you can just name your ligands, and this will automatically recognize your script file and do the calculations for you if you're using Autodoc Vina. In this, we are doing the Autodoc only. So can we do? So right now I don't have the Autodoc Vina, I'm sorry, but I can suggest you some tutorials so that we can do later. So any other question? Okay, so we'll move to the practical session. So I will be providing you this file, that is the protein ligand docking using Autodoc. So there are some stuff which is written here. So there are some important things which you need to note. And basically we'll be first preparing the molecules for the docking. Second is steps involved in molecular docking. And third one will be the analysis of the docking results. So this file will be provided. So, so first thing is downloading your molecules. First one is the protein file. So basically you can go to the RCSP PDB file, PDB website. And if you know the name, uh, either you can type in the name of your protein, or if you know the ID of the protein, that is 2RGU in this case, we'll be using this one. So you can type in the ID here, hit enter, and simply find out the protein of your choice here, right? So here you can see there are some informations. This is a crystal structure complex of human DPP4 and some inhibitor in this case. So you can find out more information. Some important information are like X-ray diffraction. This is the method by which this protein has been determined. The resolution. So lower the value, better the resolution is. That is, it is a high resolution structure. So we should prefer if the structure is having less than 3 angstrom resolution, or you can go for checking whether there are multiple entries in the database if you have there. So chances of the missing residues are less if you have a better resolution structure, right? And so many other information, especially the literature, is published here. If you want to know more about this protein, so you can always go to the PubMed or the literature resource which is given here. If you click on this link, it will show you the PubMed page for the link to the article, right? So, so this is the protein. How to download it? You can go directly and check for the PDB file format. So you can click on this one. It will download the protein for you. And simply you need to copy this into your working directory, right? So, okay, another important thing is that I have mentioned here in this file. First thing is update Python for Windows. I have attached one tutorial here. So you can see how to update, especially for Windows. And then is make sure your working directory name contains no gaps. So sometimes what we do is, for example, I have created next gen helper like this one. I create this folder for molecular docking. So uh, the Autodoc, it doesn't like it. So basically, if you are trying to separate the names, either put underscore like this, right? Or just try to make it a single string. Don't put gaps there. So Autodoc, it doesn't like it that way. So just connect the whole thing together. This is the type of name you should give to your directory in which you are going to work. And if you see, I have put it into the C drive. So again, it is basically the Autodoc will follow the path. And if it finds some gaps in that path, so there can be some errors usually. So I suggest that you work in the C drive only. In the C, you can go to this PC and then go to the C drive, make your directory here, new folder, and then start copying your materials here, right? So I will copy this. Uh, I have copied that file from the download folder and pasted it here. This is the first thing. Second thing is downloading uh, the molecule from the database. This can be any molecule, can be from any source. There are so many databases from which you can download the molecule. So the molecule name in this case is linagliptin. So what I'll do is I'll proceed to PubChem, which is one. So and put my name there, linagliptin. You can go to PubChem and then put the name. So this is the first hit here which appears. So this is linagliptin. So here you can see you have 2D image and you have a 3D conformer. So as was the question that you don't have the 3D conformer, you need to convert this 2D structure into 3D by using energy minimization protocol, right? So I'll click on this one because 3D structure is available with me. So I'll go with the 3D conformer. You can rotate and see that this is a 3D conformer for my structure. So if I want to download the 2D structure, you can also do that, and you need to convert into 3D conformer again. So I will download this SDF file here. and again copy this file into my working directory that is in C here, right? So, all right. So now my molecules are ready. So I'll go and open Autodoc, Autodoc tools, right? So it must be installed on your PC. So I'll click on this one.
Sir, which one is better, Autodoc or Autodoc Vina?
See, both have their functions. So basically, vina is used for the virtual screening of the libraries, usually where you have uh, compounds. And Autodoc is especially uh, developed for a single ligand binding. If you have a single ligand, you want to do exhaustive docking, that you can do with Autodoc uh, for. So they have their own function. You can't do the screening here until you make some script and try to do multiple dockings here with the Autodoc. So they have their own functions. Usually in virtual screening, we need to uh, reduce the exhaustiveness, and so it depends on the search algorithms also, or the scoring functions also which you use there. So in my opinion, applications are different. If you are using single molecule and checking nice conformation for or binding poses for a single molecule, go with the Autodoc. Or if you're involved in library screening, go with the Autodoc Vina in general. So uh, I have opened this Autodoc tools. So one important thing which you need to do is, first of all, set the preferences here. So what does it do? Uh, Basically, you need to connect the path of uh, this software to the working directory where you are working. That means every operations which will be carried out afterwards, it will be set into this directory only. So I'll put this set, and this window will appear. So basically, what you need to do is go to your working directory. I can simply copy this path here, right? So this is your working directory, if you remember, in the C drive. So simply click here and copy this path, and go to this setup directory section of this window and simply put the path here. Now you need to set this path and make default. That means you are setting this path by making default. You will, every time you will be logged into this particular folder. So for now we want this because we want every calculation to be done in our particular directory. So I'll dismiss this one. So it it will be confirmed once you try to read the molecule. So file, read molecule, it will directly lead you to the folder where we have the file here, right? So, okay, so there, so you have options whether you can prepare these molecules with help of Autodoc tools, but I would suggest that you proceed with UCSF Chimera. So you have to install this program also, that is UCSF Chimera. Okay, so I will open this and I'll try to prepare both my molecules, that is a protein and then ligand with the help of Chimera. So file, open, and sorry about that. Can't move this window here. Okay. So this is the PDB file or protein file which we process first. All right. So here you will see that this is the PDB file, and if you go back and check the RCSP file, so it is a dimer basically, right? So this is one monomer, this is another monomer. So for this exercise, we need the monomer only, so we can uh, go to select chain and select chain B. So we'll proceed with the chain A for a molecular docking exercise. Just that we don't have more atoms in the file, so we don't want the chain B. So I can select this one. I can go to actions, then atoms or bonds, and then proceed with the delete. So I just want to delete the molecules which are present here. So delete it, right? So it will delete your chain B. So now you have only chain A. So now you can see there are some additional atoms here. There are heteroatoms. There can be water molecules. There can be non-standard amino acids. So basically this exercise will delete the chain B and delete the non-standard residues, whatever except for protein residues that will be deleted. So I'll go to select residues and all non-standard residues. So under this non-standard residues, you can see some ligands such as NAG356 and some water molecules are available, right? So all these come under the non-standard. Everything else than the protein atoms will be considered as a non-standard in this case. So I'll click on non-standard residues, right? So only you can see non-standard are selected. So I'll go to action, atoms and bonds and delete these non-standard, right? So these are uh, deleted. So I'll try to save my file right now. Save this PDB and to the same folder. I hope it's gone to the same folder. Yeah, so I'll try to name it as protein.pdb. Save and close session, and then try to open another file. That is a conformer. This is the ligand molecule which we will prepare now, right? So this is the ligand. So for this ligand, one important thing is that you need to check for the hydrogen bonds, hydrogen atoms, if they are present. Also check for the bond orders. So this chimera can do the corrections. So basically you can go to tools, then in structure editing section, you have options like add hydrogens and add charges. So we'll be doing that and followed by the minimization. So add hydrogens. So you can proceed with, okay, please check what is happening there. So next is again tools, add the charges. So in this case, gas taker charges will be added, that is optimized for the ligands. So you can press OK. And then finally move with the structure editing with the minimizing. This is the structure optimization step. So basically, if you have obtained a 2D conformer, you can minimize the structure to obtain a 3D structure, a 3D conformer, right? So you have to do minimization here. This will give you several options. So let me bring this down. Yeah. Okay, so you can see there are several options to minimize the structure. Basically, it uses steepest descent and conjugate gradient um, minimization algorithms to perform the minimization. So uh, you can increase the steps to more if you have a larger molecule. So you need more number of steps to minimize or optimize the ligand. So maybe you can proceed with the 1000 and 100 for this conjugate gradient, right? So for now, we will proceed with the default ones and start minimizing the structure. So it will do the minimization. Basically, will form the 3D conformer of your molecule. So once the minimization is complete, you can see the dialog beneath this. This is completed. So here it will show you finished minimization like that. Okay, so then you need to save this file. So remember that you need to save the mol2 file this time. So for protein, you will be saving PDB file, and for ligand, you'll be saving the mol2 file. So you can name this as ligand and save. So let's confirm whether the files have been saved. All right, so you can see protein, and then you have the ligand molecules which are saved. So go ahead and delete the older ones so that we have only our molecules here. All right, so now we have somewhat prepared molecules, that is ligand and protein. We'll go again to MGL tools in the Autodoc. We'll again try to follow some important steps which will read the molecules in Autodoc platform. So all right. So you can always follow this guide that what are the things which are happening here. So we have followed this preparation of the protein and slight preparation of the ligand molecule. Now we'll be proceeding with the protein and ligand processing in Autodoc. So I'll go to file, read the molecule for the protein. And you can see the protein is visible in the visualizer, right? So this is the protein. You can change the different visualizations from here like this. So I'll keep it simple like that. Okay. Second one. So next step will be preparing the molecule, that is this particular protein. So we'll go to edit and make sure once again we'll do the hydrogen thing. You need to add hydrogens. So ideally we need the polar hydrogens. For this step, we'll be doing this all hydrogens, and then we'll merge the hydrogens. Go to edit hydrogens and merge non-polar hydrogens. Basically, if there are some uh, non-standard hydrogens which are present in the protein structure, they will be merged and only the polar will be shown. So it is the polar hydrogens which will often take part in interaction with the ligand. So we don't need all the hydrogens, only polar will be enough. So now you can see polar hydrogens are present in our protein, right? So this is the hydrogen step which is completed. Make sure your protein doesn't have any missing residues. Otherwise, if you have any missing residues, you can follow this step. That is, go to edit, MISC, and check for missing residues. So I'll confirm it. Go to edit, go to MISC, and check for missing atoms, you can see. So I'll click on this one. So in my case, it is 0 residues with missing atoms. But in some cases, there can be missing atoms. So you have to select all residues and follow another step, that is MISC and repairing the missing atoms. That will take some time. You have to wait. Once that is complete, you can proceed with the next step. So we have completed up to here. That is the hydrogen bond, hydrogens. Then you need to save your protein, or we can proceed with the ligand processing simultaneously, right? So I'll go to this ligand section here and I'll try to input. And here you'll see there's no match, but remember to change this section to all files. Then you'll be able to see your ligand molecule that is in mol2 file format. So open. Okay, so you'll keep the charges from the chimera which we obtained. So I'll go and search for the ligand which is there. Yeah. So this is the ligand which has appeared. It has a different, still it's not docked, so it is away from this protein. So you can see it. So there are some processing steps which are involved in this ligand processing. Now, basically, you have to find the root of this molecule. So root is basically the program recognizes that where this molecule, what is the root of this molecule, and it takes some atom as a reference and will find out uh, uh, the rotatable bonds accordingly, right? And then you have to choose the torsions, right? And then set the number of torsions. If there are more number of torsions, we'll go one by one. So I'll go to ligand. Then torsion tree and detect root. So if you see some small green colored sphere has appeared, that means this is the root of this particular molecule. And then I'll go to ligand, torsion tree, and choose torsions. So now you will see the change in color was automatic, and the molecule is mostly in the red color, and there are some green colored bonds. So these green colored bonds are the degrees of freedom or rotatable bonds. That means these bonds will be changed by the search algorithm in Autodoc. And often, uh, in this case, out of total 32, there are 6 uh, rotatable bonds. So if the molecule is large enough or exceeds more than 25 rotatable bond, it doesn't work. So you need to reduce the number of rotated bonds in that case. So for now, this is 6, so it is good enough. We'll proceed. And then we need to, if you want to set the number of rotatable bonds, you can set from here. Right now it is maximum 6, so it's acceptable. I'll put proceed. Then you need to save the pdbqt file of the ligand. So remember that this is by default going to the directory which we have selected, ligand.pdbqt. So automatically it will save that file, right? All right. So this ligand processing has been done. So protein processing is done, ligand processing is done. Next is your grid formation. This is one important parameter. So for you, I have given some X, Y, Z coordinates here, right? So X, Y, Z coordinates, they correspond to Cartesian coordinates. That means they will define the location of an atom in 3-dimensional space. So you can use these coordinates. They are particularly for this protein only. So important question is, how did I obtain these XYZ coordinates? So I'll go to Discovery Studio. There you can do it, right? So you can basically open the file which I have deleted. So basically you can import that PDB which you have downloaded. Let me go and download the PDB again. So I'll delete the B chains and the waters. So only I have obtained is this ligand, which is the co-crystal. So remember that there are three ways. One is literature, from where you can identify the residues which will form your binding site around. Second one is uh, by prediction, right? Or by the co-crystal, right? So this is the co-crystal which is bound here. So this is the binding site on this protein. So I'll use structure, monitor, and the centroid. So this will generate this centroid around this ligand. And if I right click and go to the attributes, it will give me these XYZ coordinates here, right? 50.7, 48.6, and 35.3. So these you can copy. So I have already copied it for you here. So we can use this one for defining the binding site. So basically we'll define the grid around here. We don't consider the other residues, but just around this bound ligand. This is the binding site where we want to uh, perform our molecular docking exercise on. Okay. So grid formation, we'll be following the steps one by one, right? So basically you have this grid. So the first thing is choose the macromolecule, because you have opened it already in the file in the visualizer. So you will choose from here, that is protein. So it is chosen. So just press OK. It will ask you to save the PDB-QT file for the protein. So I'll proceed and save it. All right. So next is the grid box. So basically go and choose the ligand. That is ligand, select ligand. And then you can go with the grid box formation. So here you can see there is a box which has been obtained here. So this box is randomly placed in the cavity. We don't know where this box is right now. So remember these coordinates, they will be helpful now. So you can simply put in the values there. Let me see what are the values. It is 50.74. Forty-eight point 63 and 35.37. So if I hit enter, so it changes the grid to the position around that binding site, right? So if you feel that grid size is small or large, so you can use the spacing to increase the volume as this grid in all the directions, right? So this is important step. So you need to define the grid large enough so that it can involve all the binding site residues. And secondly, it should be able to accommodate your ligand. So this one is the finalization of the grid based on the XYZ coordinates from here. So next step is file close saving current, and that's it, right? So we have to save the GPF file, that is grid parameter file. So we'll go to output and save as, I'll name it as grid dot grid parameter file, that is GPF, and save it. So basically all the parameters, the information of X, Y, Z coordinates, it will be saved in that file. Second one is next is run parameters, that how you are going to run the grid parameters and set up the parameters for your molecular docking further. So after grid, you have to go to docking, right? Go to docking, macromolecules, set the rigid frame. Here you'll be setting the rigid frame and is the protein file which you have just saved. Open it, and then you have ligand, then choose the ligand from here. So just accept, it will give you the different informations which you did, like a number of torsions at the beginning. Accept it, and then docking, then you have search algorithm. This is the genetic algorithm here, right? So this will define the exhaustiveness of your run. So how many runs you will do. That means how many number of poses you will generate for your molecule, it depends here. So number of poses, if you want to increase, you want a more exhaustive search, more number of poses for your molecule, you can increase the number of GA runs. So most of the parameters are default and optimized very well by Autodoc. So you can do is in number of runs you can change, and you can change the evaluations. That means these are internal evaluations which are being carried out. So if you choose medium, this value will be if you choose short, this value will be reduced. If you choose a long one, so this value will be even further increased. So basically you are trying to make, by keeping it long, the search a very exhaustive one. So you can proceed for the demonstration with short. Ideally, you should proceed with the long one and can generate more number of parameters. And we can keep these as default and accept. So now these search parameters have been defined. Next, we have to click the docking parameters. So you have to just accept it. And then these are the automatic Autodoc for parameters. You can keep it as such, and you'll go to output and choose the algorithm. So in this case, Lamarckian algorithm is the most commonly used and well-known algorithm for Autodoc. We'll be using this one. But if you have other preferences, you can choose from here. So we'll do here. So after this, it will ask you to save the docking file. So I'll name it docking parameter file, that is DPF, and it is saved. All right, so you have done with saving the run parameters. Now you have two experiments which will be run. One is the grid experiment, and another one is the actual docking experiment. So here you have to run AutoGrid, and this window will pop up. So important thing is that you provide the right program path for the AutoGrid experiment. So for that,